Now look at this.
What's up, about penguins? Today we're going to do topic 4.6 on the cell cycle. So cell cycle is made up of two parts. You've got interphase made up of G1, S, and G2, and then you have the mitotic phase made up of mitosis and cytokinesis. There's three parts to interphase, and the cell is going to grow throughout all three parts. So G1 is where the cell is growing and is kind of preparing to divide. This is where it gets past the G1 checkpoint to ensure that it should divide because it got the growth factor. S phase is going to be when the DNA is going to synthesize another copy. We go through DNA replication. I need 2 copies of the DNA because I'm making 2 cells. And then the G2 phase has to do with the cell is going to grow some more and it's going to make a copy of all the organelles. So prophase is the first part of mitosis. This is where the cell is going to prepare to divide by having our chromatin is going to condense by having the DNA wrap around those histones and it condenses down. The sister chromatids, the duplicate pair of chromosomes, are now going to find one another and pair up. So metaphase, our sister chromatids are going to line up on the metaphase plate. They'll line up in the middle of the cell. As you notice, there's lines coming toward those sister chromatids. Those are microtubules. The microtubules are going to come from our centrioles at the 2 poles. They're going to extend out, and the microtubule will attach to a kinetochore that will be in the centromere of our sister chromatids, basically right in the middle of our chromatids. Anaphase, our sister chromatids will be pulled apart. So the microtubules are going to depolymerize. They'll get shorter, and as they get shorter, they pull the sister chromatids apart. There's also an enzyme called separase that's going to allow the sister chromatids to separate because it basically breaks that cohesion that's between the 2 sister chromatids. Telophase is going to be the last phase of mitosis. This is where I've now formed my 2 new nuclei. There's a nuclear envelope that surrounds the 2 nuclei, and then our chromosomes are going to decondense down back into chromatin. Psychosis happens after mitosis. This is going to split the cytoplasm, so I now form my 2 daughter cells. This is going to occur differently in our animal cells versus our plant cells. Animal cells, you're going to see there's going to be an actin ring. Actin is a type of microfilament that's going to get smaller and smaller and smaller until finally the contractile ring closes and it pops. We have 2 cells. Cell plates will be deposited due to vesicles that will drop off the pieces, form that cell plate that will then form a divider between the 2 cells in a plant cell. So in case you didn't catch it, I gotta dance. Prophase, metaphase, anaphase, telophase, cytokinesis. Yeah, sorry about that one. Anyway, so there are checkpoints that are going to take place from the cell cycle. So the G1 checkpoint is going to ensure that I should divide. Do I have the growth factor? If it doesn't get the signal, it's going to exit the cell cycle and go into this G0. So like your non-dividing cells, like your liver cells, your nervous cells, your muscle cells will go through this phase. At the end of G2, we have another checkpoint. This is to ensure that I have replicated all the chromosomes and I'm ready to divide. M checkpoint makes sure that all the microtubular tasks can occur so you can evenly divide the cell.